Imagine walking across a frozen landscape that has remained untouched for over 40,000 years. The ice beneath your feet is so ancient that it was formed when woolly mammoths roamed the earth, when human ancestors were just beginning their first experiments with fire and art, when the world was gripped by an ice age so severe that sea levels had dropped hundreds of feet. Beneath that ice lies a treasure more valuable than any gold or jewel ever discovered. It is a library of life itself. Frozen in the permafrost are the remains of plants and animals that vanished millennia ago, preserved in perfect condition by the relentless cold. And now, in a stunning, so well preserved that it is rewriting everything we thought we knew about how long biological material can survive, and what it can still reveal. The story of DNA in ice begins with a simple observation that scientists have known for centuries. Extreme cold preserves things. The frozen tombs of Arctic permafrost have yielded mummified animals that look so lifelike that archaeologists initially mistook them for remains from just a few decades old, only to discover through radiocarbon dating that they had been frozen for 20, 30, or even 40,000 years. Their fur was still intact. Their organs were recognizable. In some cases, scientists could see what the animal had eaten for its last meal, preserved in its frozen stomach. But for most of the 20th century, no one realized that the DNA itself was surviving in these remains. They assumed that over millennia, radiation from the environment and chemical degradation would inevitably destroy the delicate molecular structures that encode genetic information. The very idea that DNA could persist intact for tens of thousands of years seemed to violate the laws of chemistry. Then, in the 1980s, a revolutionary discovery changed everything. Scientists extracted DNA from mosquitoes and other insects preserved. In amber, fossilized tree resin that had sealed these creatures in a perfect time capsule for over 100 million years. The DNA was fragmented and damaged, but it was recoverable. Suddenly, paleontologists began to wonder, if DNA could survive millions of years in amber, how long might it persist in other conditions? What if permafrost, with its extreme cold and isolation from oxygen and light, was an even better preservative than amber? The question ignited a new field of science, paleogenomics, the study of ancient genomes, and researchers quickly realized they were sitting on top of one of the greatest untapped archives of ancient biological information in the world. The turning point came in the early 2000s when scientists began recovering DNA from permafrost sediments. These were not DNA from individual frozen organisms, but rather environmental DNA, genetic fragments suspended in the soil and ice layers accumulated over millennia. It was like finding pages from thousands of books scattered throughout a massive ancient library. Each page contained tiny snippets of genetic code from plants, animals, fungi, and microorganisms that had lived and died in that location over tens of thousands of years. By analyzing these fragments, scientists could reconstruct an entire ancient ecosystem without finding a single complete fossil. They could determine which species had lived together, how the climate had changed over time, and how organisms had responded to those changes. It was a method so powerful and so elegant that it seemed almost like reading the mind of the past. But the truly astonishing discoveries came when researchers focused on extracting DNA not from environmental samples, but from the remains of individual organisms frozen in permafrost. A team at the University of Copenhagen, led by a paleogeneticist named Heske Willerslev, made a startling observation. When they extracted DNA from the hair of a woolly mammoth that had been frozen for 4,000 years, they found that the genetic material was in remarkably good condition. Better condition, in fact, than DNA extracted from bones of the same age. The hair had protected the DNA somehow. But more importantly, they realized that the permafrost was providing preservation conditions so favorable that DNA survival rates were orders of magnitude better than anyone had predicted. In some cases, DNA fragments from permafrost samples were actually showing less degradation than DNA from museum specimens that had been stored in conventional freezers for just a few decades. It seemed impossible, yet the data was undeniable. The reason for this extraordinary preservation has to do with the unique properties of permafrost itself. Permafrost is not simply frozen soil, it is an environment locked in a state of near-perfect stasis. The temperature remains below freezing year-round, often reaching negative 20 or 30 degrees Celsius, cold enough to essentially halt most chemical processes that degrade DNA. Oxygen is excluded, preventing oxidative damage. The environment is sealed from environmental contamination. Microorganisms that would normally decompose biological material cannot function in such extreme cold. It is as if the permafrost creates a molecular suspension chamber pausing time itself. Organisms that died tens of thousands of years ago are preserved in a state that is almost impossible to achieve in the laboratory. They are stored in conditions so stable that DNA molecules remain largely intact, their base pairs still bonded together, their structure recognizable even after 40 millennia of entombment. What makes this discovery even more profound is the realization that as climate change accelerates, vast regions of permafrost that have remained frozen since the last ice age are now beginning to thaw. The permafrost is not a permanent feature of Earth's geography. It is conditional on cold. As global temperatures rise, the permafrost line is retreating northward. Regions that have been frozen for thousands of years are melting. And in those melting regions, scientists are finding intact remains of animals that have not seen daylight since before human civilization emerged. A woolly mammoth, so well preserved that its muscles still retain their color. A saber-toothed cat, its teeth still sharp, its body still recognizable as predator. An ancient horse, its coat of fur still visible, its hooves intact, and crucially, their DNA is still there, more complete and better preserved than anyone had thought possible. One of the most remarkable discoveries involved the remains of a wolf pup that had been frozen in permafrost in northeastern Siberia. When researchers excavated the specimen, they found an animal so perfectly preserved that it seemed as though it had simply fallen asleep a few years earlier. Radiocarbon dating revealed that this wolf pup had died 57,000 years ago, during the last glacial maximum, when ice sheets covered much of the northern hemisphere and the climate was far colder and drier than today. The scientists extracted DNA from the pup's remains and sequenced its entire genome. What they recovered was not just genetic information, but a time capsule of an extinct ecosystem. The wolf's DNA provided clues about its diet, its health, its evolutionary history. But more importantly, the presence of the wolf itself revealed information about the climate and landscape of Siberia 57,000 years ago. This was an animal that had adapted to specific conditions. Its presence and the condition of its remains told scientists about a world that had vanished. As these discoveries accumulated, paleogeneticists realized they had stumbled upon something unexpected. The conventional wisdom had been that ancient DNA degrades in a predictable, linear way. The older the sample, the more degraded But the data from permafrost samples was suggesting something far stranger. Yes, DNA did degrade over time, but the rate of degradation was not linear. It followed a different pattern. In some cases, DNA from permafrost samples that were 40,000 years old showed less damage than DNA from samples that were only 10,000 years old. The reason had to do with the specific conditions in which each sample had been preserved. Some permafrost regions were more stable than others. Some had experienced warming events that had thawed and refrozen the soil, introducing oxygen and microorganisms that accelerated degradation. Others had remained in perfect frozen stasis. By analyzing the pattern of DNA damage, scientists could actually reconstruct the thermal history of permafrost regions, determining not just the average temperature, but the specific warming events that had occurred over tens of thousands of years. One scientist at the forefront of this research is Carina Keir, a paleogeneticist at the Globe Institute at the University of Copenhagen. Keir became fascinated by the question of how long DNA could actually survive in nature. She began by examining permafrost samples from all across the Arctic, looking for patterns in DNA preservation. What she discovered was startling. In some permafrost cores extracted from regions in Siberia, she found DNA fragments from species that had gone extinct 20,000 years ago, interspersed with DNA from species that had vanished 50,000 years ago. By analyzing the vertical layers of the permafrost, she could create a timeline of the ecosystem, reading off the different organisms that had lived and died in that location, like the rings of a tree. Some cores showed clear evidence of climate shifts. A layer might be rich with DNA from cold-adapted plants and animals, while just above it, a layer showed DNA from species adapted to warmer conditions. These transitions marked ancient climate changes, recorded not in ice cores, but in the genetic code of organisms. The implications of this work extended far beyond academic curiosity. By extracting and analyzing environmental DNA from permafrost, scientists could begin to understand how past ecosystems had responded to climate change. During the last ice age, when global temperatures plummeted and much of the northern hemisphere was covered in ice and tundra, species either migrated to follow suitable climate zones, adapted to the new conditions, or went extinct. The DNA preserved in permafrost was recording which organisms survived, which evolved new adaptations, respond to rapid climate shifts. Today, as we face unprecedented warming from human-caused climate change, understanding how species responded to past climate fluctuations could provide insights into how they might respond in the future. One fascinating example emerged from permafrost cores collected in the Yukon Territory of Canada. Scientists analyzing these cores found DNA from woolly mammoths, horses, and giant ground sloths that had all lived in the same region during the Ice Age. But as they moved up through the permafrost layers examining progressively younger samples, they found the DNA signatures changing. The mammoth DNA gradually disappeared. The horse DNA diminished. The composition of the ecosystem shifted. This transition occurred around 12,000 years ago, coinciding with the end of the last ice age and the beginning of the current interglacial period. The permafrost had recorded a mass extinction event, written in the language of ancient DNA. By comparing the genetic information from different time periods, scientists could see that the megafauna extinction was not instantaneous. It was a process that unfolded over hundreds and thousands of years as climate warmed, vegetation changed, and the animals either adapted or retreated or perished. Year, or sometimes just a season of snow accumulation. By examining the isotopes and chemicals in ice cores, researchers can determine how warm or cold it was, how much rain or snow fell, even what the air contained. But only recently have scientists realized that ice cores also contain DNA. Frozen in the ice are fragments of genetic material from microorganisms, plants, and animals that were blown into the atmosphere by wind, carried by water, or became airborne during volcanic eruptions. Over thousands of years, these genetic fragments accumulated in the ice, creating a record of ancient life. A team led by researcher Celia Sykes at the University of Washington extracted and analyzed DNA from ice cores removed from a Greenland glacier. What they found astonished them. The ice contained DNA from plants that had grown in the far north tens of thousands of years ago. By analyzing the specific genetic variants in this plant DNA, they could determine which plant species had been present during each climate period. They could see the shifts in vegetation as climate changed. They could identify plants that had gone extinct and plants that had migrated. This was a level of detail that had never been possible before. Ice cores had previously told us about atmospheric conditions, but they had not directly told us what organisms were living in the regions around the ice sheet. Now they could. The technical challenges involved in extracting and analyzing DNA from ice and permafrost proved to be immense. The DNA is fragmented into tiny pieces, rarely longer than 100 base pairs. It is chemically damaged, with errors introduced throughout the sequence. It is contaminated with modern DNA from the scientists handling it, from environmental microorganisms, from dust in the air. The extraction process itself must be conducted in facilities with virtually no exposure to modern genetic material, using protective equipment and sterilization protocols that rival those used to handle dangerous biological agents. A single speck of skin from a researcher could contaminate an entire sample, introducing false data that could lead to incorrect conclusions. The work requires painstaking attention to detail, redundancy at every step, and constant vigilance against contamination. One researcher who has devoted his career to perfecting these extraction techniques is Love Delane, a paleogeneticist at the Center for Paleogenetics in Stockholm. Delane has worked on some of the most ambitious permafrost DNA projects, including efforts to recover and sequence the genomes of extinct species. He describes the experience of working with ancient DNA as a form of archaeology conducted at the molecular level. You are looking for tiny threads of genetic information in a haystack of contamination and degradation. When you find a true piece of ancient DNA, it feels like discovery in the most profound sense. You are holding in your hands a genetic message from a world that ended thousands of years ago. One of Delane's most famous projects involved recovering the complete genome of a woolly mammoth that had been frozen in Siberian permafrost for approximately 4,000 years. This was a landmark achievement in paleogenomics by comparing the mammoth genome to the genomes of modern elephants. Delane's team could identify the specific genetic changes that had allowed mammoths to survive in the extreme cold of the Ice Age. They found genes related to thermoregulation, to the development of thick fur, to tolerance for low oxygen levels at high altitude. They discovered that mammoths had unique versions of hemoglobin, the protein that carries oxygen in blood, which allowed them to function efficiently in the cold, thin air of high-altitude tundra. They even found evidence of genetic changes related to circadian rhythm, suggesting that mammoths had adapted their internal clocks to the extreme seasonal variations in daylight at high northern latitudes, where in summer the sun never sets, and in winter it never rises. But the most remarkable finding was this. When Delane's team compared the mammoth genome to modern elephant genomes, they found that the genetic differences were surprisingly small. Mammoths and elephants shared over 99% of their DNA. The genetic changes that distinguished mammoths, while significant enough to make them a distinct species, were actually quite subtle. This raised a provocative possibility. If the genetic blueprint for a mammoth had survived in permafrost ice for 4,000 years, and if scientists could identify the specific genetic changes needed to turn an elephant into a mammoth, could they theoretically recreate a woolly mammoth using genetic engineering? Could they reverse extinction? This question has captivated the imagination of scientists and the public alike. Multiple research teams have begun exploring the feasibility of what is sometimes called de-extinction through genetic engineering. The concept would be to take the complete genome of an extinct species recovered from permafrost, identify the genetic differences between that extinct species and a modern living relative, and then use gene editing techniques like CRISPR to introduce those changes into the genome of a living animal. In theory, the result would be an animal, genetically indistinguishable from the extinct species. In practice, the challenges are staggering. You would need to introduce potentially thousands of genetic changes into an organism, each one precisely targeted. You would need to grow this modified organism in a womb, raising questions about which species would serve as a surrogate. You would need to monitor the health of the resulting animal, watching for unexpected genetic interactions or problems that might arise from the artificial modifications. And crucially, you would need to create an entire population, not just a single individual, because a lone organism cannot survive. Yet despite these challenges, research is advancing. A company called Colossal Biosciences, founded by geneticist George Church and entrepreneur Ben Lamb, has announced that they are working on resurrecting the woolly mammoth. Their long-term goal is to restore populations of mammoths to the Arctic tundra, where they believe that mammoth-like megafauna could play a crucial ecological role in combating climate change. Mammoth herds, the theory goes, would trample forests and prevent tree growth, maintaining the tundra as grassland and permafrost. Grasslands reflect more sunlight back into space than forests, and permafrost stores vast quantities of carbon. By restoring mammoths, the thinking goes, we could help preserve permafrost and reduce climate change. It is an ambitious vision, one that raises profound questions about humanity's relationship with nature and extinction. But it is only possible because of the remarkable preservation of DNA in ice. The discovery of intact DNA in permafrost has also provided insights into the health and living conditions of ancient animals. Scientists can extract microbiota DNA from frozen intestines, determining what an animal ate and which microorganisms lived in its digestive system. They can analyze parasites preserved in the feces, understanding which infections afflicted ancient creatures. They can examine the chemical composition of ancient bones, determining nutritional status. In one remarkable case, a woolly rhinoceros, found in permafrost in northeastern Siberia, was analyzed so thoroughly that scientists could create an almost complete medical history of the animal. They found that it had suffered from parasitic infections, that it had been healthy overall, that it had survived a volcanic eruption during its lifetime, as evidenced by volcanic ash particles in its teeth. This ancient animal, dead for 34,000 years, became not just a specimen, but a subject of biography. The implications of permafrost DNA preservation extend to understanding human prehistory as well. Several human remains have been recovered from permafrost, and their DNA has revealed information about ancient human populations. In Siberia, a boy have suffered. The permafrost had preserved not just bones, but hair and skin, allowing direct examination of tissues. The DNA extracted from this child told stories about a population of humans that had lived during the transition from the Ice Age to the modern climate. Understanding how human populations had adapted to climate change in the past provided crucial context for understanding how modern humans might adapt to future changes. But perhaps the most urgent question raised by ice-preserved DNA concerns the fate of the permafrost itself. As climate change accelerates and temperatures rise in the Arctic at rates twice as fast as the global average, vast regions of permafrost are beginning to thaw. When permafrost thaws, the genetic information that survived thousands of years of freezing can be lost forever. The discovery of DNA preserved in ice has also transformed our understanding of ancient climate change. By analyzing DNA preserved in ice cores in permafrost, scientists can now see how organisms responded to past climate fluctuations. They can see that when climate changed in the past, organisms did not simply shift their ranges. Many evolutionary changes occurred. Species that survived climate changes were not randomly distributed populations, but specific lineages with genetic adaptations to the new conditions. This suggests that future climate change will not simply move species north or to higher elevations. It will create evolutionary pressures that will select for populations with genetic traits suited to new conditions. Some species will evolve rapidly, others will go extinct. The permafrost DNA record shows us that extinction was a common response to rapid climate change in the past. As permafrost continues to thaw due to climate change, it will release not just DNA and microorganisms, but also methane and carbon dioxide that has been sequestered in frozen ground for thousands of years. This will accelerate climate warming, creating a feedback loop. As the permafrost thaws, it releases greenhouse gases, which warms the climate further, which causes more permafrost to thaw. The discovery of ancient DNA preserved in this permafrost has given us a window into what will be lost if this process continues unchecked. The permafrost is not just a storage facility for genetic information. It is a carbon sink, an archive of ancient life, and a time capsule containing evidence of how ecosystems responded to climate change in the past. Some scientists have proposed more radical solutions. If permafrost DNA contains the genetic blueprints of extinct species, and if de-extinction becomes feasible, could we use this technology to restore lost species to the Arctic ecosystem? Could we bring back not just woolly mammoths, but also extinct horses, woolly rhinos, and other megafauna that vanished at the end of the last ice age? And if we did, what would be the ecological consequences? Would restored megafauna herds actually help preserve permafrost and combat climate change, as some theorists have suggested? Or would reintroducing extinct species into modern ecosystems create unforeseen problems and unintended consequences? The DNA hidden in ice has given us tools to answer questions we never thought to ask about the past. It has shown us that extinction is not always final, that genetic information can survive incomprehensible spans of time, that the permafrost is not a barren wasteland, but a library containing the secrets of ancient ecosystems. As we face the challenges of climate change and the potential loss of modern species, the lessons hidden in ice become ever more relevant. The permafrost is teaching us that genetic diversity, evolutionary adaptation, and ecological resilience are fragile. It is showing us that climate change has reshaped ecosystems in the past, with devastating consequences for some species and transformative effects for others. And most importantly, it is reminding us that the present moment is crucial. The permafrost is melting. The genetic archive is being erased. The time to preserve this information, to learn its lessons, and to make choices about the future based on the wisdom of the past is rapidly running out. Every year that passes, every additional degree of warming, every new thaw event brings us closer to a point where the DNA hidden in ice will be lost forever, taking with it secrets that may have been crucial to our survival and our understanding of life itself.